The thing we're going to discuss today is basically to have a review for our practical. You know, the practical is coming next week, and it will be available for you starting Wednesday, May 6th. Let me check my calendar here. It will be Wednesday, May 6th, all the till Friday, May 8th. So I will have this practical open for you from Wednesday till Friday. And as you know, this practical is basically going to cover 6 units, as you know, and those are diffusion, Unit 8 enzymes, Unit 9 cellular respiration, Unit 10 photosynthesis, 11 cell cycle, 12 biotechnology. Those will be covered in this. So to make this kind of review efficient, let me start first. I already have, I gave you a test, I remember a few days ago, and let me start with this test. So let's go through this test together and see if you have any question. Hello, Devin, how are you? Good?
Very good.
Good, great. Okay, so let me share with you my screen. Okay, let me share my screen with you, and I will go first. Let's go with the last quiz about meiosis and biotechnology. And let me go through this with you very quickly. Okay, now can you see the screen?
Yes.
Yeah, I can see it.
Okay, great. So the first question was basically talk about cell division and especially meiosis. And when I talk about meiosis, you need to know the stage, different stages of meiosis. You need to know, the end product of meiosis. So when I talk meiosis, it means that I start with one cell, I end up with four cells. And I start with a diploid cell, meaning that each chromosome has 2 copies, and I ended up with 1 copy per cell, okay? Which is a reduction in the number of chromosomes. So that question here about anaphase. When you talk about anaphase, you are talking about separation, or getting the chromosomes or sister chromatids separated from each other. So in meiosis 1 and meiosis 2, there are differences. So in meiosis 1, what is separating is basically homologous chromosome. So homologous chromosomes separate in meiosis 1, okay? And then that question was, what about meiosis 2? So meiosis 2, what is going to separate is the sister chromatids, okay? Remember this.
So meiosis 2 is like
So yeah, let
mitosis?
me do that. I have some people, let me do. So remember this again. Meiosis 1, meiosis in general can be divided into 2 different phases, meiosis 1 and meiosis 2, which are basically almost identical except for very major things happening in each phase. Remember in meiosis 1, when you talk about anaphase, the homologous chromosomes separate. On the other hand, if you talk about meiosis 2 during anaphase, they are sister chromatids separate. Don't forget this, okay? And now there is a different kind of cell cycle, we call it mitosis. And the question here was, if you start with a cell and that cell has 24 chromosomes, okay, and the cell undergo mitosis. So when you talk about mitosis, cell is basically each cell gives 2, not 4 like meiosis, 2. And each cell will have same number of chromosomes exactly like the starting cell. So this is another very important difference. In meiosis, you end up with 4 cells. Each cell has half the number of the chromosomes. in mitosis, you end up with two cells and they have the same number of chromosomes like the parental or the original cell. So in this case, if a cell has 24 chromosomes undergo mitosis, so you expect to see two cells, okay? So it is two cells, so it's either this answer or this answer. And how many chromosomes will be exactly the same number of chromosomes, no change. So the right answer here is going to be two cells. Each cell has 24 chromosomes, okay? Here it says how many cells are produced from a cell performing meiotic. So again, for meiosis, we are expecting to see 4 cells, okay? Question 5, during which phase of the cell cycle the DNA copy itself? This is very important. So remember, the DNA copy itself during the interphase and especially during the S phase. S stands for synthesis, meaning the DNA is replicating, is copying itself. So this happened, it is not happening in prophase, not in metaphase. You know, it happened only in the interphase, especially during the S phase, okay? And here it says which statement correctly describes difference between meiosis and mitosis. The main difference between the two is gametes only produced during meiosis. Gametes are only produced during meiosis, okay? Here's a chromosome line up on a metaphase plate only in mitosis. This is wrong because it line up in meiosis and mitosis. Total number of chromosomes is reduced on only in mitosis. This is wrong statement because the total number of chromosomes reduced only in meiosis, okay? When mitosis occur only in prokaryotes, there is no meiosis or mitosis in prokaryotes, simply because you don't have nucleus. We don't have nuclei. So the right answer is going to be meiosis, always associated with gamete production. Here it was a figure, and I ask you, to tell me the different phases in this. That was onion skin showing cell division. So A for sure represent anaphase. When I say anaphase, this is a separation of the chromosomes or the sister chromatids in this case. So sister chromatids are separated during anaphase. Here, this represent prophase, where there is no mitotic cell division yet. What happened here is this is the nucleus, this is the cell wall, and you can see in the nucleus is basically the nuclear membrane is kind of degraded, chromosomes kind of condensed, okay? So it's typical for prophase. In this case, all chromosomes are aligned in the center. So this is middle or metaphase. So you expect to see this in your practical. I will have several kind of picture showing several stages of cell division, and it will ask you what stage the figure or the picture represent. So always remember, cells, chromosomes are in the middle, this is metaphase. Chromosomes or sister chromatids separated like this is anaphase. Nucleus with degraded nuclear membrane, condensed chromosomes, this is prophase, okay? Here, when you see cells like that, where there is no condensation of the nucleic acid, so this is represent an interphase. I'm not going to confuse you with this, but you will always see this kind of phases in your test. Like here, this is also anaphase. This is another anaphase here. What is the difference? This is different timing. So if I ask you which one happened first, this one happened first because the sister chromatids is still kind of close to each other. Here, they are kind of separated further from each other. So this is very important in your practical. DNA paternity testing. First of all, you are going to see this picture for sure in your practical, okay? This represents an agarose gel, and those represent the well where we load the DNA And each band represent a DNA fragment, okay? So each one of those represent a DNA fragment, and the separation here is based on size, meaning that smaller DNA molecules will run faster, so we'll see them at the end. Larger DNA molecules or fragments will run slower, so you see them at the beginning. When we apply electricity, this is very, very important to know this. For separation to happen, you need to apply some electricity. We apply volt from minus to plus, from minus to plus. Why? Because DNA is negatively charged. So when you apply negative, what happens is it is going to repel, and that will force the DNA fragments to get and to move further from the charge. So they're going to move toward the plus. Okay, so always negative, this is where I load the DNA. Positive, this is where the DNA is migrating to. So this is one of the most important thing you need to know about electrophoresis. Before I proceed, do you have any question for particular, like gel electrophoresis in general? So any question for me regarding gel electrophoresis in general?
I do not have any questions right now.
Okay, anybody else?
No, I'm good.
Okay, so now when you see a picture like this, it will ask you, like, here, this is DNA from mother, this is a DNA from a kid, and those are DNA from different, like, males, and we need to assign the kid, to one of those males, okay? It's paternity testing, and it depends on what we call DNA fingerprinting. And DNA fingerprinting is basically to see those DNA fragments that the kid has in common with his mother and his father. Remember, 50% of the DNA for the kid come from mother and 50% come from the dad, from father. So when you see a picture like this and it asks you which is one of those four males, F1, F2, F3, F4, one of those is the father, biological dad for the kid. So you need to look, first of all, at the DNA fingerprint of the kid. I can see 6 DNA fragments, 1, 2, 3, 4, 5, 6, okay? This DNA fragment, for example, it is common with the mother. So I can't consider, I can't take this for comparison. So that DNA, it looks to me, since it shared with the mother, so it probably came from his mother. Come to this DNA fragment. So it is not coming from his mother because you can't see anything here. So it should come from his dad. Now, if you look at the picture, you can see two fathers here. F1 and F4 have common DNA fragment with the kid. So now I can point to either father number 1 or father number 4 to be the dad for this kid. So I go further, again, another DNA fragment the kid has shared with his mother. So I can't put this in consideration. The same here, a DNA fragment similar to the DNA from his mother. I can't consider it as well. Now go further, I will see a band here. So this band, there is nothing in his mother, so it should come from his dad. So if I look carefully here, I can see either father 3 or father 4. So now at this point, there are two DNA fragments came from father 4. That one and that one. In this case, I can circle father 4 to be the biological dad for the kid. So the question here was two different kinds. One of them was how many DNA fragments are shown in the kid DNA sample. You simply count. Each one of those represent a DNA fragment. 1, 2, 3, 4, 5, 6. And it asks which is the biological dad for the kid. So definitely it will be number 4 here. Do you have any question for me for this part? how to interpret like a gel for paternity testing or for crime scene investigation. What you need to know that each band here, this line represents a DNA fragment, okay? And you need to know that the DNA, when they run through a gel, they run from minus to plus. Why? Because DNA is negatively charged. And also you need to know separation is based on size. Smaller DNA fragment will run faster. So it will be at the end. Larger will run slower. So it will be at the beginning. Okay? Shall we proceed?
Yep, I think you explained that pretty well.
Okay, I'm happy to hear this. Anybody have any question for this part? Because this is important. You will see this absolutely in your practical. Okay, now go to the last question here, and the last question asks about blood typing. And blood typing, when I do blood typing, you know, what I'm doing, as a matter of fact, I'm trying to find an antigen on the red blood cells. Okay, blood cell type A, they have antigen A on the surface. Blood type B, they have antigen B. Blood type AB, they have both antigen A and antigen B. Blood type O, they don't have any antigen, so the cell has no antigen. Okay, so when I do blood testing, how I know this? How I can detect the antigen so I can specify the blood type? To be able to do this, you need to use a specific antibody, known antibody. Okay, so for example, for antigen A, I need to use antibody A, an antibody that was generated against antigen A. For antigen B, I need to use antibody B. For antigen AB, you know, I can just add both. That's it. It's not going to hurt. Okay. There is another group, blood group, we call it RH, and RH is another antigen. It's completely different from antigen A, antigen B. Okay, also the way to be able to detect it is using antibody against that particular antigen. So when you do blood testing, you take a blood from, say, person X, okay? And you have those kind of well. So you take a drop of blood in plate number 1, plate number 2, plate number 3. Just blood, red blood cells, regular blood. And then now you need to detect the antigen. So the best way to detect the antigen, you use antibodies. So say I'm going to use antibody for antigen A and mix. Antibody for antigen B and mix. Antibody for antigen RH and mix. And then look for this kind of particle formation. Particle formation and aggregation will tell me there is a reaction. Okay? So here, for example, there is no reaction, meaning that antigen A is not present. Why? Because antibody A did not react. So when antigen A is not present, so this one is not blood type A. Okay, look here. There is a reaction when I added antibody B, meaning that antigen B is present. Okay, when a cell has antigen B, that is blood type B. So that is blood type B. Now here, it will tell me if this blood type has an RH factor or no. A reaction happened here. So it means that it is plus. We call it plus. So this blood type here is going to be B plus, B plus. It is not going to be A because there is no reaction. If it gives me the same, like when I added anti-RH, it gives me no reaction, so it will be B negative or B minus. Okay, you got this one?
Okay, that cleared up a lot of things for me. Thank you.
Yeah, no problem. Let me, I have also here another Let me do this. I will share with you this presentation just to make it kind of, this is a complicated slide, but it will tell you a lot. Okay, so remember blood type A, we have 4 different kind of blood type in human. Okay, we call those A, B, O blood typing. And those can be blood type O, blood type A, blood type B, or blood type AB, 4 different kind. Okay, and the thing that make those blood different is basically the red blood cells everybody has. Okay, so our red blood cells, they have a specific kind of sugar associated with it. And that specific kind of sugar will tell which blood type we are. Blood type O, for example, they have only the precursor sugar, those 4, like it's like a oligosaccharides made of 4 monosaccharides only. Okay, and this is common in all blood type. All blood type have this common, we call it the precursor. Everybody has this. If you are O or A or B or AB, your blood cells have this precursor here. So what is the difference? If you go to blood type A, you have an extra sugar here, N-acetyl galactosamine. If you have this extra sugar, so you are A. Okay, if you have a different sugar, which is galactose only, not N-acetyl galactosamine, you are B. If you have both sugar, now you are AB. Okay, so this is what is the antigen that I was telling you about. Those are simple sugar that can be used as marker, and it can tell you the blood type of a person. How I know that blood type A has this? Simply, if I have antibody against this one. So if I have antibody, I add antibody against N-acetyl galactosamine, it will react. And when they react, they will do coagulation and precipitation. If I add like antibody B, it is not going to react. Okay, it is not going to react. This is why in human, for example, if you are blood type A, so you have this kind of sugar, in your plasma, you don't have antibody for this. Otherwise, you will die. What you have antibody for a different blood type. In this case, like blood type B. You have in your plasma, you have red blood cells, you have serum. So your serum has antibody against different blood, not against your own blood. So if somebody like a blood type A get blood transfusion from B, what's going to happen? As soon as you introduce red blood cells to A, the recipient, the antibody, which is anti-B in the recipient blood, is going to react with the antigen on the donor red blood cells. That would cause kind of clotting, and that can cause to lead to heart failure, strokes, and all those bad things. Okay, so this is the basics for blood typing. What you need to understand, you don't need to worry about all this mess. When you do blood typing, you use known antibody to detect unknown antigen. Okay, you use known antibody to detect unknown antigen, and that will tell you about the blood typing. Okay, so that was for blood typing, so remember this. In this slide, remember again, when we run DNA, we run electricity from minus to plus, So we load the lanes. Okay, DNA is negatively charged and it's going to separate based on size. So it was going to, there will be if there is the same charge, so that will force the DNA in the well to move toward the plus. Okay, this is the most important thing about unit 12. Other things that you need to really know for this unit, when you talk about DNA isolation, we added No, unfortunately, haven't done this in the lab. I'm so sorry for what happened. It's out of our hand. But you need to know there are reagent added and they have different function. So if we try to extract DNA from the nucleus, we need to add those reagents. And you will be asked what is the function of each one of those. So amide tenderizer, this is like a protease. A protease meaning an enzyme that destroy proteins, including enzymes, including those enzymes that attack the DNA, like DNases. You need to get rid of those. Otherwise, they will attack the DNA and you will degrade it. You need to add another reagent, a detergent, like soap. Detergent because it interferes with the plasma membrane and nuclear envelope. So it will facilitate the release of the DNA. Also, you need to know that ethanol, ice cold, 95% ethanol was added to precipitate the DNA. So this is the function. If you have DNA solution and you need to get it out of solution, you add ethanol. So those are three things, three reagents. You need to know the function of those in this unit. This is the main or the most important thing about this unit. Do you have any question so far?
No, I'm all good.
You got okay. So let me
That's on
see
me.
here if I can go. Yeah, let me do this. There was also, I gave you like extra credit assignment, I believe for this unit. Let me go through this quickly with you. So I'm sharing this with you. Can you see it? Can you see this extra credit assignment?
Nope.
No, I
All I
can't.
see is the Zoom page.
Oh, you can see it? Okay, let me do this.
Okay, we can see it.
Yeah,
Can
no.
you see it right now?
Mm-hmm.
Okay, great. So here, that was a question regarding enzymes, okay? And I need from you, because it is very important, probably you will see the same in your practical. When you look at a graph, you need to be able to, if you are here, this graph is basically the correlation between the rate of the reaction or activity of the enzyme on the y-axis and the temperature used in the x-axis, okay? So if you look at this graph A, for example, you can see enzyme activity is very close to zero almost, okay? No activity at all at around 0 degrees Celsius, and then the activity is going to increase significantly when we increase the temperature. Here we call this the optimum temperature, and this is the best temperature for the enzyme to give the best product, okay? And then when we increase the temperature further, the activity is going to slow down, slow till it reach at 50 degree, almost zero activity again, okay? So here the question is about an enzyme, and that enzyme is basically to make pigments for cats, okay? Siamese cats. And it says that the enzyme is basically to make the pigmentation, and that enzyme is thermolabile, meaning it had high temperature will kill it, will make it inactive, okay? So it says here, if the enzyme will be like at high temperature, that will kill that enzyme and there will be no pigment at all, okay? So if you have rabbits, for example, or Siamese cat or whatever, and you try to develop those like at 5 degree, this is where the enzyme is going to be active. So you are expecting to see lots of black pigmentation. When you increase the temperature a little bit, so they noticed or they realized that the the animal was mainly white, but a little black, like around the paws and stuff like that. When they increase it further to 35 degree, no pigmentation found at all. So it say here clearly which graph in those represent this, or the description. An enzyme which is going to be active around 5 degree, and then less active at 20, and then it will be completely inactive around 35. So if you look at this here in graph A, that tells you that the maximum that enzyme was very active, like around 5, 10, 11, 12, 13. And then when you go to 20, the activity started to decline. And then when you went to 35, for example, it even declined more. Okay, so this graph here, this is the best graph to describe this paragraph here. So the right answer here should be graph A. So as I mentioned, I need from you, when you look at a graph like this, to be able to, based on the correlation, to tell about the optimum activity of an enzyme. Optimum activity meaning the maximum product that can be produced. And here in this case, see, maximum product here. So if you drop like a line, that will give you that the optimum temperature for this enzyme is 13. Same deal here, it is a different enzyme. Maximum activity is observed around maybe temperature like 40. Because if you draw a line, you will reach around, you will intersect with the x-axis around 40. So 40 degrees is the optimum. In this case here, you can see roughly it's maybe around 30 degrees, okay? This is why when I said, what is the optimum temperature for enzyme shown in graph B? So this is graph B. And again, optimum temperature, I can see roughly it's around maybe from 83 to 40. So here is the choice, okay? Is it 35, is it 45 or 50? So it is for sure, it is not 50, okay? Is it 35? 35 is somewhere here. So it is not 35. So it will be this chance, it's a 45, okay? That was another question here. It says, when you do, if you have a dye and you do some kind of diffusion, which is basically movement from high concentration to low concentration. And this is a simple calculation here. It says that the dye diffuses like 2 centimeters per minute. So how long it will take if it moves 1 decimeter? A decimeter, if you remember, is 10 centimeters. A decimeter is 10 centimeters. So going from 2 to 10, this is 5 times. So you need to multiply this times 5. So if it moves 2 centimeters per minute, it will take 5 minutes to move 1 decimeter. And how can we make this process proceed or speed up? Remember the factors affecting diffusion. One of them, temperature. Increasing the temperature a little bit will increase the diffusion rate. So you just write 5 in this, and increasing temperature. Okay, and then you submit your quiz. When you look at this, don't worry, because every single time I do like blank like this, I do manually, because I know that your answer, even my answer C, is different a little bit this time. And this kind of different wording will consider it wrong. But don't worry. I don't need for you to email me and say, oh, I believe I did this wrong. No, don't worry. My job, after you finish the quiz, I go through this and make sure that your answer is matching what I need. It shouldn't be completely matching 100% with this, okay? So don't worry about losing any grades in this regard. Okay, so now it's your turn. You need to ask me what kind of question you have. Hello. You still there? Did you left? Did you already left?
No. How many questions in total is going to be?
Okay, let me tell you what was the test or the quiz is going to be for 1406. This is 30% of your grade. Okay, so 100 point. For 1408, it is going to be 20% of your grade. And the number of questions is going to be 50. Okay, and it will be very, very similar to practical number 1. Again, multiple choice, true or false. I'm going to give you pictures and graph, and I will ask you a question about this. It's going to be available for you, like I will open it Wednesday and it will be closed on Friday. So you have this window of time. You can take it anytime you like. Before I proceed, Unit 7 all the way to Unit 12, what is the one that you feel it was the most difficult and you need from me to go through some of it with you now?
Okay.
Unit 7 was diffusion, Unit 8 was enzymes, Unit 9 was cell respiration, Unit 10 was photosynthesis, 11 was cell cycle, meiosis and mitosis, and 12 was biotechnology.
Meiosis and mitosis was the one that got me.
Meiosis and mitosis. Let me see here.
There was just a lot of terms that I kept getting mixed up between the two.
Okay, let me see here. So I need to select. How can I do this? Okay, can you see the, no, not that one, no, I'm sorry. Okay, what you see now on your screen?
I see DNA isolation.
Oh, okay, let me,
From here.
no, I need to stop sharing this and I need to share a new one. Okay, can you see the, this, yeah, like Unit 11 in your screen?
Yes, chromosome replication. Okay, unit 11, cell division.
Okay, good. Can you see it?
Yes.
Okay, great. What you need to know from this, as I mentioned at the beginning of this review, you need to know the difference between mitosis and meiosis. Okay, so mitosis, the main idea for mitosis is basically to increase in number, increase in size. And each cell, when it divides, it gives two identical cells. Identical, meaning they are the same exactly, having same number of chromosomes. Okay, meiosis, on the other hand, it gives 4 cells not identical. And this is due to what we call homologous recombination. And what else? It gives cells half the number of chromosomes in each one. We call this haploid. In human, for example, human, we have different kind of cells. We have cells that undergo mitosis, like our liver cell, our heart cell, our muscle, to increase in number, increase in size. And we have other cells that undergo meiosis, like our gonads, those cells that produce like sperms and eggs. Okay, those cells are haploid. So in your liver cell, you have 46 chromosome. Each cell has 46 chromosome. The sperm or the egg, they have 23 chromosomes. So they have half the number. And we call this haploid, okay? So when you look at the cell cycle, you need to know that there is a big, huge phase, and this is most of the time the cell is going to be within this phase, which is interphase. An interphase by itself can be divided into 3 subphases, G1, S, G2. What you need to know from this, that in S phase, this is the only phase where DNA is replicating. So when the cell is about to divide, they should replicate its DNA. And this happened first. So before the nucleus starts to divide, the cell grow and multiply the number of chromosomes. We call this the S phase, or the synthesis phase, where DNA is happening. You remember this, you need to remember this. And it happens in interphase. The second phase, we call it M, or meiotic or mitotic, M phase. And this is where the nucleus is dividing. And then the third, we call it cytokinesis, or this is where the cytoplasm is dividing, okay? What else you need to know here? Chromosomes. When there is no cell division, so the cell is basically metabolically active, producing enzymes, lots of gene expression is happening. Chromosome is not condensed. Chromosome is basically present in a form, we call it chromatin. Chromatin, like thread, big, huge, long thread wrapped around proteins. We call this chromatin, okay? And then when the cell receives a signal to divide, so the DNA is going to replicate, meaning each chromosome is giving 2 copies, okay? And the DNA is going to condense very tightly. Now we call this chromosome. So remember the difference between chromatin and chromosome. Chromosome is more condensed structure. Chromosome this is when the DNA is replicating 2 copies. Chromosome made of 2 identical copy, we call those sister chromatids. Sister chromatids. So what are sister chromatids? Those are 2 copies of the chromosome, but they still attach via centromere. So they are not individual chromosome yet. They are going to be individual or completely independent when they separate from each other. Nor they are sisters, still young, lived in the same house together. Later, they will separate and each one of those will go to a different house. You imagine it like that, okay? So we call those sister chromatids. Those are identical sequence. Each one of those is made of double-stranded DNA, okay? Double-stranded DNA, double-stranded DNA, and those are identical. So looking carefully here, the genetic material is basically going to double. And since they are doubled here, they need to get separated again during cell division because they can't keep doubling like that. You need to know the different phases. I don't need to go through each one of those, but remember the name PMAT. PMAT. P for prophase, M for metaphase, A for anaphase, T for telophase, PMAT. And you start with like sister chromatids attached like that in prophase. Okay, you can see all those like nuclear envelope is kind of perforated, kind of degraded a little bit because it's going to disappear, allowing only the chromosome to start separating. Okay, spindle fiber are going to form. Those are very, very important for mechanical separation of sister chromatids. Those are made of microtubules. So you are expect that the microtubules are going to assemble to make spindle fibers like that. All this during prophase. During metaphase or the middle, this is all the chromosomes are aligned at the middle. And everybody is basically attached to spindle fiber and to the centrosome, centrioles, ready for the big action, the big move, where sister chromatids are going to separate. Remember, this is a very important difference between meiosis and mitosis here. And I need for me to write this down. in mitosis during metaphase, chromosomes are aligned side by side. Okay? Chromosomes are aligned side by side. In meiosis 1 especially, during metaphase, homologous chromosomes are going to face each other. They are not going to align. They're going to be facing each other because anaphase is going to follow. And those chromosomes, homologous chromosomes, are going to separate during meiosis. During mitosis, sister chromatids separate. Okay? So anaphase meaning apart from each other. This is where sister chromatids separate, and that will follow up with the telophase. Telophase meaning two nuclei are going to form. Okay, so now each sister chromatid, now it starts to be like decondensed and will be chromatin again. Okay, ready for another metabolic activity. Spindle fiber, there is no need to keep those, so they are going to degrade and recycle. Nuclear envelope will start to form. And after that, you end mitosis with cytokinesis, where the cytoplasm is going to divide. Now we have two cells instead of one, identical, because chromosomes basically replicate, give the same copy exactly, having same number of chromosomes. So that was for mitosis. So any question for mitosis before I jump to meiosis?
Nope, you explained a lot of the misconceptions I was having with that.
I'm really glad to hear this, Gregory. Thank you very much. Anybody else have any question for me?
No, I'm all good.
You good? I'm pleased to know that, man. You did very good job. As a matter of fact, you know, you guys are like fighters, because this kind of condition is not easy, especially when you talk about biology, you know, that you need to do things with, you know, with your hand, and all of a sudden you are taking online virtual biology, which is kind of hard. I know that for sure. But you till now you are doing your best. I am glad, you know, and just to give you a hint, I will be with you till the end after you finish your practical, and if I see you struggling going from one like grade level to another, if you are in a like in the merge of like 2-3 point, not 2-3%, please, because sometimes I receive emails that, you know, I still have 2 points to go from B to A, but what she mean or what he mean is 2%. When you say 2 points, it's 2 points. 2% meaning maybe 30 points or 25 points. That's will be hard for me to do this or override this. But if you know you're 5 points, say 7 points away, so I can, you know, give you a small assignment to do, like in 24 hours, so I can jump you from one grade level to another. I will do that. If it is 2 points only, I will give this. You know, I will not hold you in a B for 2 points, okay, or 2.5 points. But if you are like 5-6 points away, I will give you an assignment to do, quick, easy, so you can get those extra points to jump you from one level to another. So I will do my best for you, but what you need to do your best, and no much left, you know. That remind me about the lab report. Still, some students didn't send it. It's a big chunk of points. So if you haven't done it yet, try very much like this weekend to finish it and email it to me, because I need to have a window of time to go through it and grade it. Okay, so you are going to expect to see something like this, like skin cell, onion skin, and different kind of cells, and the point like a pointer, and it will tell you to not to describe, but you need to mention the phase. So you need to study those very well. You need to know the feature for each phase: prophase, metaphase, anaphase, and telophase. Meiosis is kind of different a little bit, because in meiosis you are not worried about increasing in size what you need to produce more cells. And those cells are gametes. And those cells should have the number of chromosomes. We call those haploids. And those cells shouldn't be identical. If you look at human being now, everybody is different due to this kind of what happened during gamete production. Lots of recombination is happening. It happens only during meiosis. Your liver or your heart is, don't worry, it is now the same heart, is the same liver, so you don't need to change. So it is just increase in number. It's basically cells already died, need to be renewed. Okay, your liver is not looking for a new look, basically. But human race, we always look for different kind of property. So we do this during gamete production, which is meiosis. In meiosis, again, it is PMAT. Again, meiosis can be meiosis 1 and meiosis 2. Meiosis 2, you consider it exactly like mitosis. You don't need to worry about it. Meiosis 2 will be exactly the same like mitosis. All the act and all the fun is during meiosis 1. What is going on in meiosis 1? In meiosis 1, especially in the prophase, this is where DNA is basically already replicated, condensed. Two things are happening in prophase 1, not in prophase 2, just prophase 1. Synapses and crossing over. So synapses is going to set the stage for crossing over. Synapses meaning that homologous chromosomes that you have is going to pair. Remember, we have each cell we have 46 chromosomes, okay? Those is 23 pairs. 22 pair is basically homologous, comes from dad, have come from dad, have come from mother. We call those homologous because they are very, very, very similar. They are not identical though, because your mother and your father are not the same. So their genetic makeup is different. But they have chromosome 1, mother have chromosome 1, father have chromosome 1, and you will have 2 copies of those chromosomes in your cells. Those, we call those homologous. So those homologous chromosomes are going to synapse or pair. That will set the stage for crossing over or exchanging of some genes. Some segments between homologous chromosomes is going to exchange during this phase. Only happen during prophase. Metaphase 1 is different from mitosis because, as you can see, homologous chromosomes are aligned in the center and facing each other. So that red and yellow, those are homologous chromosomes facing each other. That green and blue, they are homologous facing each other. If you look carefully, you can see that the yellow has a small piece of the red, and the red chromosome has a small piece of the yellow. Why this? Because during the synapses and crossing over, so some genetic material already transferred from between homologous chromosomes. Anaphase, normal, those chromosomes going to separate. And then telophase, sometime it happens where nucleus is going to form. In some cases, that anaphase, it will follow up with telophase, no nucleus at all, and then meiosis 2 will start. And in meiosis 2, now the chromosomes are going to align side by side during metaphase, exactly like mitosis. Sister chromatids are going to separate exactly like mitosis. And the end product, as you can see, 4 cells. Those are not identical because they shared some pieces of the DNA between mother or maternal chromosome and paternal chromosome. And they have the number of the chromosome, so they are haploid. Is the main thing about meiosis and mitosis. So the bottom line, you need to remember BMAT. You need to know what is the special for each phase. You need to know that only during meiosis 1, the homologous recombination, synapses is happening, especially during prophase 1. You need to know during anaphase 1, homologous chromosome separate. You need to know during anaphase 2, sister chromatids separate. And you need to know the number of the cell products. Which one give you haploid, which one give you diploid. Okay,
Okay, thank you.
well, no problem. You have any question, guys? Anybody else?
No.
Great, I'm glad. Any unit need that you really need to go through before we finish this?
I'm trying to see which of the quizzes that I did terrible on.
I believe cell respiration was one of the most terrible, and photosynthesis. Let me see here. Let me see here. Let me go to, I think Unit 8 is easy, especially you did the lab report, so it shouldn't be that hard.
Let's see, cellular respiration. All right, I was on Pearson, so let's go to that.
Okay, so now I have, this is the cell respiration unit. Can you see it in your screen?
Yeah. Yes.
Okay, great. Again, as I mentioned in my last Zoom presentation, what you need to know from this unit, we have cell respiration in 3 different models: animal, plant, and yeast. For animal, I need from you to know 3 things. I need from you to know the unit to measure metabolic rate. Metabolic rate here in this experiment, basically with the amount of oxygen consumed. Okay, and the unit is mill oxygen per hour per gram. You need to remember this. This is the way to measure a metabolic rate for an organism, by knowing how much oxygen is consumed per unit time, which is 1 hour per mass. So that will tell you about the metabolic rate of the individual or the animal that you are examining. Number 2, you need to know that this is very true for warm-blooded animal, that the smaller the animal, the higher the metabolic rate, the larger the animal, the slower the metabolic rate. So if you compare hamster with elephant, hamster very, very tiny with very, very high metabolic rate. Elephant so big with very low metabolic rate. So you need to remember this as well. Also, during this virtual experiment to measure oxygen consumption, we have in this respiration chamber, we have potassium hydroxide pellet. So that is, we call it the white pellet. And what is its function? Is basically to absorb carbon dioxide and convert it to to potassium carbonate. So it is not going to interfere with the oxygen, because you are going to measure oxygen only. The animal is taking oxygen and producing carbon dioxide. So now you have gas which made of 2 different kind of materials. So you don't need carbon dioxide to interfere with oxygen. So you need to get it out of the equation. The way you do this, by adding potassium hydroxide. Okay, so this is the function of potassium hydroxide in the respiration chamber. Those are the 3 things that you need to know from this experiment. Unit for metabolic rate, the animal, the metabolic rate, the correlation between metabolic rate and the size of the animal, and finally, this potassium hydroxide, the name, you need to know the name and what the function. The second model was peas, okay? And remember, we used germinated peas. One set was boiled, so to kill it, to inactivate or deactivate all the enzyme. Okay, so basically preventing cell respiration. and another set you left it without boiling. And he was measuring, not measuring, he was detecting carbon dioxide in this experiment. And how we detect carbon dioxide? By using a reagent we call it phenol red. You need to remember the name. Phenol red is an indicator that detects carbon dioxide. If there is any carbon dioxide in solution, the solution is going to be acidic. Phenol red is going to change color to yellow. If there is no carbon dioxide, phenol red will stay red. Okay, so in this experiment, you have your independent variable. The variable that you manipulate was basically to get your peas boiled or non-boiled. What depend on this? Respiration, carbon dioxide release, color change. This is also what you need to know from this experiment. So if I give you two different test tubes, and I said that what was the reagent used, you need to tell me phenol red. When I say which tube of those contain like carbon dioxide, you look at the color and you can tell this is yellowish color, golden, this is red. So mean that this one is basically that has carbon dioxide indication for cellular respiration. Okay. For the yeast and fermentation experiment, you need to know the name of the device, which is a fermentation tube. Okay. In this fermentation tube, what we supposed to do is basically we need to test different kind of sugar and see which sugar work the best for yeast. And an indication for this is going to be the amount of carbon dioxide released. That will tell you, if the sugar was used efficiently or no. And this is a quantitative experiment because you can measure. Here you are not detecting carbon dioxide with phenol red. Here you are measuring it. So basically you mix your yeast. You mix it with the sugar, different kind of sugar, and you let it stay at room temperature, say for 40 minutes. After 40 minutes, if the yeast is utilizing the sugar, it's going to produce carbon dioxide. Carbon dioxide is going to, since everything is closed, so carbon dioxide is going to push and it will accumulate like at the tip of the tube. And since it is graded like this, graduated, so you can measure and tell how much carbon dioxide was produced. And that's the thing that you need to know from this experiment. What else? You don't need to worry about the structure of ATP. You need to know that the equation for cellular respiration is basically the degradation of organic compound to release energy. Okay, and in order to be able to break down organic compound to release energy stored here, you need to have oxygen. So oxygen is like the fuel that you need to add to burn and break down this organic compound. When you break it down to carbon dioxide, basically. Okay, what is the end product? Energy stored here is going to be released in a form of, we call it adenosine triphosphate. That is, the cell can use this. So the cell, the only currency that the cell can utilize, it will be energy in the form of ATP. And that is the main idea from this unit. No more, no less. Okay. Question for me.
Let's see.
That made sense to me, but
Great. If anything else, any unit that you need to go through quickly?
I'm looking right now. I'm actually pretty good on most everything.
same here.
Good. Glad to know that.
Maybe a refresher on diffusion and osmosis since it's been a while.
I
But
know that is the most difficult one.
but other than that, I'm pretty much good.
Okay, let me see here what I have. Um, okay, let me see. Osmosis. I need to go back and I need to share. It was here somewhere. Okay, let me see. Okay, can you see Unit 7?
Yes.
Okay, great. Unit 7 is hard. I agree with you. Unit 7, you'll see also lots of questions for Unit 7, cell diffusion, diffusion and osmosis. First of all, you need to know the difference between diffusion and osmosis. And when I say diffusion here, we're talking about components or materials that diffuse freely, okay, from high concentration to low concentration.
Mhm.
Okay, and diffusion here depends on many factors. First of all, it depends on the size of the molecule. Small molecules diffuse faster than heavier molecule. Adding energy will also expedite and increase the diffusion rate. So if you look at this experiment here, there was two different kind of dye used, and the main difference between the two dyes here, potassium permanganate and Janus green, is the size. Potassium permanganate is 294 Dalton versus 511 Dalton for Janus green. So you can see roughly Janus green is twice the size, the molecular size like as potassium. So if you look at the experiment, we have Janus green in lane A and the potassium permanganate in B, and we let that plate, is agar media, we let that at two different kind of temperature. One was at room temperature, one at was 35 degree, to see the effect of the temperature. And here is an example. After 30 minutes, you can see if you measure the diameter, you can see definitely that solution B diffuses much faster than solution A, based on the rate, the size of the ring or this old halo that form around the well. Okay, that was a very simple experiment. So if I give you this and ask you which solution diffuse faster, or which solution has a smaller molecular weight, you need to tell me that solution here has less molecular weight because it diffuses faster. Okay, and also you need to know that temperature has an effect. If you increase the temperature, even the diffusion rate will be more faster. You need to know the difference between a solid, a solvent, and a solution. If you have a cup and you have a spoon of sugar, sugar here is going to be your solute. If you dissolve it in water, water is going to be your solvent. The two together will mix and make a solution. Okay? And you need to know solution can be divided into three different kinds based on salt concentration. Those with high salt, we call those hypertonic. Those with low salt, we call those hypotonic. And this was like same salt, isotonic. But I will not be able, if I give you, for example, I put 1 gram sucrose in 100 ml water, and I give you 101% sugar, and I said, okay, Gregory, can you tell me 1% sucrose? Is it going to be hypotonic or hypertonic or isotonic? What you are going to tell me?
Uh, can you say that again? I was having a brain fart.
Yeah, that's okay. I have a question for you here. I gave you a solution made of 1% sucrose. So I have 1 gram sucrose in 100 ml water, and I told you, can you tell me the solution is hypotonic or hypertonic or isotonic? Can you answer this?
No, because there's no salt.
No, it has 1%. It has 1 gram salt in
Oh, I thought you
100
said
ml.
sucrose.
Yeah, so yeah, okay, that's a good point. When you say salt or solute, you know, it can be any kind of material which is like a powder like that. That
Oh,
can be
okay.
sodium chloride, that can be sucrose, that can be glucose, that can be calcium chloride. So we call those solute. Okay.
Okay,
So
thank you.
my mistake when I say salt, yeah, I should say solute. Okay. So if you have a solution made of 1% sucrose, is it a hypotonic or a hypertonic or isotonic?
Are we having anything to compare it to, or
Exactly, you are absolutely right. Now, this is if I ask you this question, you need to ask me again. You say, I need to compare it with something to be able to tell. I can't tell you it's hypo or hyper or iso unless I have another solution to compare. So when you look at those, you are comparing two solutions or two states together to be able to tell, okay, which one is hypo, which one is hyper, which one is iso. So here is for you, I have a beaker. I have two different solutions, 10% sucrose, 50% sucrose. And they are separated by a membrane which is semi-permeable and that allow only for water to diffuse. First of all, if before allowing water to diffuse here, two solutions, which one of those is hypotonic, which one is hypertonic?
the 50% is hypertonic, while the 10% is hypotonic?
Perfect. So this is hyper, this is hypo. This has more salt or more solute than this. Great. Now, when you look at the diffusion, which is water movement now, okay? Water movement. Water move from high water concentration to low water concentration. In this case, what do you think? Water is going to move from where to where?
It'll move from the 10% to the 50% because it is a much higher water concentration.
Absolutely. Here, 10% sucrose, meaning approximately 10 grams sucrose, 90 ml water. Here you have 50 grams sucrose, approximately 50 ml water. So 90 ml, 50 ml. So which one has more water concentration? This one. So water is, if I allow this to move, only water movement, so water is going to move toward the high salt concentration. And definitely what's going to happen, the concentration is going to increase, the volume is going to increase till it reach equilibrium, meaning that the salt concentration or solute concentration will be the same. We call this equilibrium, okay? So here you need to know from this unit the effect of different kind of solution on living cells. Names you need to remember. When I talk about plant, there are two terms that you need to be familiar with: turgidity and plasmolysis. Those only associated with plant cells. If you put plant in a hypotonic solution, hypotonic, meaning you are over watering your plant. You have lots of water in the soil, okay? You have more salt concentration inside the cell than the soil. So water is moving into the cell, and that will make the cell increase in size. We call this turgidity, okay? The cell is not going to rupture or break down because they have a very tough, rough cell wall. It's going to protect the cell. Plasmolysis is the opposite. You have your tomato plant, you put it in soil, and by mistake, you decided to add some fertilizers, but you added too much fertilizers, too much salt on the soil. So what happened is this high salt concentration is going to drag all the water from inside the cell out. So the cell is going to shrink. So the plasma membrane, excuse me, is going to detach from the cell wall. We call this plasmolysis. Here in animal cell, situation is different. In hypotonic solution, there is no cell wall. Cell will increase in size and it will rupture, lyse. On the other hand, if you put animal cell in hypertonic, so water is coming out, cell is going to shrink. We call this shriveling, cremination, no plasmolysis. Plasmolysis, plant, shriveling animal. You need to remember this. Very good example. You will see this definitely in your practical, and it will ask you, this is Elodia placed in different solution. So which solution is hypotonic, which solution is hyper? So in hypotonic, cell is turgid, inflated, as you can see, nice and healthy, smiling to you. Here, the cell is basically shriveling. The plasma membrane is basically detaching from the cell wall. So we call this plasmolysis. So this is hypertonic solution. This is hypotonic solution. In this solution, solute concentration in outside the cell is more than inside. So water is coming out. Okay. You imagine this as solute as magnet that attract water. Whenever there is lots of magnet, water is moving toward the magnet. So this will make you not confused about this. The second experiment was the effect of the molecule size on diffusion rate. And you need to know from this experiment different reagent, the name of the reagent, and why we use those to detect what, and what is the end product. Silver nitrate to detect chloride ion. If there is chloride ion, it will give you whitish cloudy color. Barium chloride to detect sulfate ion. Benedict, as you remember, to detect sugar glucose, reducing sugar. Iodine to detect starch. Biuret to detect protein. Remember those. That experiment was basically easy. We have a dialysis tubing and we have a mixture of this reagent here. Sodium chloride with sodium sulfate and glucose and starch and protein together. It's a mix. And as you can tell, they are different in sizes. This is the smallest, this is the largest, okay? And we put those in a dialysis tubing and we put it in a beaker with water and we let it like this for 40 minutes. So what is going to happen is basically molecules going to diffuse from high concentration to low concentration. But there will be one condition. If the size of the membrane allowing, if the size of the membrane is large enough to allow those molecules to come out. So after 40 minutes, you took aliquots from here and you add the reagent and you detect and see which one of those was able to diffuse out from the dialysis tubing. That was the main experiment. So what you need from to know this experiment is basically reagent used to detect what material and what it looked like if it is positive, what it looks like if it is negative. Okay.
Okay.
I missed you, lost you somewhere?
You explained it pretty well.
Anybody else, guys? You got it?
Yeah.
Okay, I'm
Yeah, I
glad.
got it.
Okay, good luck. Just study hard and you will be good, okay? Do your best and you will be great.
Thank you
No
Thank you.
problem.
for your help
Thank you.
today.
No problem, no problem. I hope this helped. I hope so.
Yeah, this is really appreciated.
No problem. Thank you very much,
There's
guys.
a lot of my questions.
You have anybody any question for me before we finish?
I think I'm good.
Yeah, I'm
Okay.
good.
Okay,
Same
great.
here.
Great. So good luck. So the test, the practical will be available for you starting Wednesday. Okay.
All right.
Okay.
Perfect.
Okay.
Thank
Okay.
you.
Thank you,
Have a
sir.
good rest of your night.
You too. Thank you very much. Bye bye. Bye bye.
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 Okay. Bye bye, guys. Bye bye. Thank you very much. Bye bye.